Bismillah ar-Rahman ar-Rahim. I would like to welcome my dear students today to talk about the histology of bones. We shall talk about the bone cells. We shall talk about the bone types, cancerous and compact bones. We shall talk about the bone surfaces, the processes of ossification and bone growth, and the factors affecting bones. As we know that bones are specialized, hard, calcified connective tissue, which is rich in calcium phosphate salts in the form of hydroxyapatite crystals. The function of bone include support, protection, homeostatic regulation of blood calcium and formation of blood cells, as well as muscle attachment. We know that bone is one of the connective tissue types, which is composed of cells and extracellular matrix, like all other types of connective tissues. The cells of bone are osteocytes, osteoblasts, and osteoclasts. The extracellular matrix is mineralized and consists of calcium phosphate in the form of hydroxyapatite crystal. So the bone matrix contains type 1 collagen, proteoglycans, and glycoproteins, and hydroxyapatite crystals. We have to know that hydroxyapatite crystals, together with collagen fibers, are responsible for hardness of bones. The bone cells may be osteocytes, osteoblasts, osteogenic cells, or osteoclasts. We shall talk about each one of these in the next slides. This is number one, the osteoprogenitor cell. These are mesenchymal stem cells, in fact, found in the periosteum or lining Haversian canals. If you don't know Haversian canals, we shall talk about it soon in the next slides. They also becomes transformed later into or differentiated into osteoblasts. The osteoblasts are the cells building the bones. They also secrete bone matrix and have a vital role in growth and repair. The next cell type is the osteoblast. The osteoblast are responsible for bone calcification and formation of matrix. These cells are polygonal or cuboidal in shape, and you see this bluish or basophilic cytoplasm. These cells, you can find it on the surfaces of bone, on the Industrium, and lining the Haversian canals. They also synthesize and maintain the matrix of bone. Later on, they can differentiate into osteocytes, the basic cell type of bone that help in maintenance and survival of bones in general. They also secrete the osteoid, synthesize the osteoid, which is a newly made uncalcified matrix of bone. This figure shows some osteoblasts here with basophilic cytoplasm. These cells help in the growth and formation and repair of bones, in fact. The other type of cell are the osteocytes. These are unique cells, in fact. They are living inside special spaces which are called lacunae. Lacunae are similar to those found in cartilage. We talked about them in the lecture of cartilage. These, in fact, are not empty spaces. They contain nourishment fluid that help support these cells. The cells usually have uh, thin extensions, or thin filaments which are running inside lacunae, inside the canaliculi. These thin extensions are called canaliculi. The canaliculi help to distribute signals and nourishment from one place to the other inside the bones. Okay, so let's see this image of osteocyte. You see these canaliculi, which are the thin extensions running from one cell reaching other cells. In fact, the osteocytes are, as we said, are living or surrounded by lacunae. Lacunae are the spaces similar to those of cartilage. The osteocytes help in maintaining the matrix. They also communicate by gap junctions, which make a society of bones communicate with each other, forming a syncytium, a society that transporting signals, transporting nourishment, minerals, water from one cell to the other. We have to know that osteocytes are essential for bone survival and matrix maintenance of matrix. The death of osteocyte will lead to bone resorption, rarefaction, or what is called osteoporosis. Osteocytes also are smart cells that act as mechanoreceptors, detecting the mechanical load on bones to trigger the activity of osteoblast and osteoclast. We shall talk about osteoclast soon. This is a larger image of the previous one. You see this osteocytes here communicating with the neighbor one via these small channels, the canaliculi. The cytoplasmic extensions run in these thin channels, reaching other cells and communicating with them via gap junctions. Okay, this is an electron micrograph of an osteocyte. You see this cytoplasmic extensions running in, and this is the here the lacunae surrounding or around the osteocyte. The last cell type is the osteoclast. The osteoclasts are generally large, multinucleated cells with branched and cytoplasm or branched cells. We are having a ruffled border. You see this ruffled? This is what is called the ruffled border. Um, they are also maybe found, they are usually found in what is called Hauschep's lacunae, a special confined space in the industrium of bone. The industrium is the inner surface of bones facing the bone marrow. We can find the osteoclast residing there in the Hauschep's lacunae. The cells are acidophilic. They have acidophilic cytoplasm with many lysosomes and many mitochondria. Osteoclasts may actually arise from the monocytes, like other macrophages of connective tissue. They arise from the connective tissue they are from the monocytes of the blood. Osteoclast activity is controlled by cytokine called osteoclast stimulating factor, which stimulate this cell to start their activity and absorb or destroy the matrix of bone and release the calcium content into the blood. This is actually the action of parathyroid hormone, which stimulate their activity, while the calcitonin act oppositely. It inhibits the activity of osteoclast. The osteoclast belong to the mononuclear phagocytic system, which actually comes from the monocytes, and their function is to release the lysosomes into the extracellular space and act as a phagocytic and remodeling cell. Remodeling help in the shaping of cells, the shaping of the matrix and the shaping of bone to grow and adapt a special shape and size. This is the osteoclast here, found in the Haversian Lacunae and facing the bone marrow here. In these are the bone marrow cells, and this line is the industrium. This is the industrial surface of bone. Let's talk about the bone, the long bones. This is the epiphysis, and this is the diaphysis here, the long, and this one is the metaphysis between the diaphysis and the epiphysis. And the inner cavity is called the medullary cavity. Okay? So let's talk about the types of bones. Bones may be cancellous or spongy, or being compact or dense or cortical bone. So they are either cancellous or spongy. Actually, they are not spongy like a sponge. They are not soft like a sponge, but they contain many spaces like a sponge. But both types of bones are, of course, hard bones. They are hard. There are no soft bones like sponge. It is called spongy since it holds multiple irregular cavities like a sponge. But the other type is compact and contains few or minimal amount of spaces or cavities inside. Type. The compact bone is also called lamellar or cortical bone. They have a solid structure and found basically in the shaft of long bones. This is a compact bone. We see here the Haversian system. Haversian system consists of multiple osteons. This is called an osteon. The osteon, in fact, consists of a central canal, which is the Haversian Canal. The Haversian Canal is surrounded by multiple lamellae of osteocytes. These are osteocytes forming lamellae, the lamellae of a compact bone. Okay, this is a nice diagram of a compact bone, 3D figure, to see the lamellae and the Haversian system of a compact bone. You see here the industrium is the inner surface which faces the bone marrow, while this is the outer surface which is covering the bone. The periosteum is the analog of the perichondrium, we have talked about the perichondrium in the cartilage lecture. And this one is the covering of bone similar to that of the cartilage. Okay? Here we see an H&E section of a bone on the right side here, a decalcified bone here. You see, this is the nuclei of the osteocytes, and this is the Haversian Canal, and this is the osteon here, osteon plus osteon. We have forgot to mention the transverse channels connecting two Haversian canals. Let's switch to a pointer pen. You see, this is a Haversian canal, and this is another one. There are transverse canals connecting the two channels together transversely, which is called the Volkman Canals. Okay, and this one is another preparation on the left side here. You can see the osteocytes arranged in lamellae surrounding the HC or Haversian canals. Okay, the Volkman and Haversian canals, you have to know that Haversian canals here contain blood vessels and contain nerves here inside them. Also, they may be lined with osteoblasts and osteoprogenitor cells here in the line in their lining. Okay? Right. Haversian canals, see the contents here are clear. They may be nerves and vessels like vein and arteries, small vessels, of course, running inside these Haversian canals. The other type of bone are the spongy or the cancellous bone. They, as I said, they are not soft like a sponge, but they contain multiple irregular spaces like a sponge, but they are hard, of course. There are multiple trabeculae forming a network or a meshwork. The spaces of this meshwork are continuous and interconnected by cavities. They are located, they may be found in the skull, the sternum, pelvis, and the ends of long bones. Okay. Here is the H&E preparation, the decalcified preparation of the cancellous bone. We cannot find the Haversian system here. You will find irregular bars like this one, irregular bars, anastomosing and branching, irregular islands of bones containing here on the edges contain osteoblasts here, and inside they contain, you can see the nuclei of osteocytes. And here is the calcified matrix, the calcified matrix of bones. The spongy bones, okay, the spaces in the spongy bone contain bone marrow, like different from the compact bone. In the case of compact bone, they are found in the long bones like this. So the cavity is continuous along the length of the bone. But here, the spaces in the spongy bone are irregular. They are anastomosing and branching freely inside the cancellous or the spongy bone. This is an H&E preparation of a spongy bone. You can see the irregular bone cavities here and here, the bone marrow cavities. And this is the industrium of the spongy bone. Actually, you can find a lot of osteoblast here lining the industrium. And inside the solid parts, here the hard, the matrix, you can find the nuclei of the osteocytes. And now let's talk about the surfaces of bones. On the outer side of bone, if this is a bone like this, generally speaking, so the outer membrane is called the periosteum, similar to the perichondrium. Here we say ostium instead of chondrium. We say the periosteum is the membrane lining the bones. Similar to the perichondrium, if you remember, it has an outer fibrous layer and inner cellular layer. But here they contain osteoprogenitor cells. In the outer part of the periosteum contain much fibers in the periosteum. Here, the outer part contain collagen fibers, which may give rise to, which help or facilitate the attachment of muscles via Sharpie's fibers. So Sharpie's fibers are used to attach muscles to the periosteum, giving them a strong attachment to bones. The bones also have an articular surface. If you look at, say, the whatever, a synovial joint like this, see here, two long bones articulating via a synovial joint. At this area, the ends of bones are covered by what is called articular surface. This surface is covered by a hyaline cartilage. And this cartilage, as we said in the cartilage lecture, is not covered by a perichondrium. The endosteum is the third surface of bone, which is found in the internal surfaces of bones, like this. It may be regular or irregular according to the type of bone. As we said, in compact or cancellous bone, there are different types of in the shape of endosteum, maybe regular or irregular cavity. The inner lining of compact or spongy bone facing the medullary cavity. Let's see this nice figure to view the periosteum. You can find the outer fibrous periosteum, the outer fibrous layer, and this is the inner osteogenic periosteum, which contains the osteoblasts here in this area, osteoblasts. And you can find deeper, you can find the compact bone here with the Haversian canals here, surrounded by lamellae of osteocytes. The medullary cavity is the space which enclose or surround the bone marrow. The bone marrow is found in the medullary cavity, and it contains what? Contains developing blood cells, network of reticular fibers and cells, red marrow. It may be a red marrow or yellow marrow. The bone marrow may be red marrow if it is active. If it is forming blood cells, it is called red bone marrow. while if it is a resting bone marrow, not functioning, it contains much more fat cells, so it is called yellow bone marrow because of the fat cells. Bone histogenesis is the process of bone formation. There are two main types of bone histogenesis. It is either an intramembranous ossification or endochondral ossification. The intramembranous one occurs in a layer of condensed mesenchyme, okay? And the endochondral depends on the presence of cartilage, so it occurs within a cartilaginous template. The intramembranous ossification occurs mainly in the flat bones, like the skull and the clavicles. It starts with a mesenchymal tissue where the cell differentiate into osteoblasts, forming primary ossification centers. Here, islands of developing bone are formed like spicules, and finally, there will be fusion of the spicules forming irregular bars of bones or the spongy bone. Look at this diagram. Here in the step number 1, you can see the center of ossification here in the intramembranous ossification. You'll find the mesenchymal stem cells, then forming what is called the osteoid around them. Then later on, the bone matrix, the osteoid, is secreted within the fibrous membrane, collecting the cells around the center, gradually differentiating to the next one. Step number 3, you will find the spicules of bone. The osteoid is anastomosing, coming and touching each other, forming irregular spaces. Finally, the bars here are contacting each other, forming the spongy bones. Okay, but in the endochondral ossification, the other type, you will find the primary ossification, which begins in the diaphysis. In case of primary ossification, you will find this is actually in primary ossification. This is the first bone formed in the long bones. You'll see in the next diagram. We'll watch this clearly in the diagram. Later on let's switch to the diagram. I think it's much useful. Look at this diagram, and this is the step number 1 here. The primary ossification center is formed here, where the first bone is formed, and it is called the primary bone, or by primary ossification center. And around the edges here, we can find what is called the bone collar. A membrane is formed along the two sides of the bone surrounding the outer surface. It's called the bone collar. And gradually, this center forms irregular bars like a spongy bone. And gradually, it becomes elongated, forming a long bone and leaving terminal parts, the epiphyseal cartilage, remain at the end of the two bones. Here at the two ends, there will be another center for bone formation, which is the epiphyseal bone growth. Let's see the epiphyseal bone growth, or the epiphyseal cartilage. At the epiphyseal cartilage, you can see the secondary ossification center, which contain several zones of growth forming the bones. Look at this diagram. This is actually a photomicrograph at the end of the growing bone to see the secondary ossification center. The secondary ossification here is another type of intra-cartilaginous ossification. Here the growth actually goes on inside the epiphyseal ends of long bones during childhood and after puberty. It grows by the effect of growth hormone and other hormones to on the epiphysis. It stimulates the epiphysis to start proliferating. The cells here, the cartilage cells, will start proliferating, forming rows of growing, of proliferating cartilage cells here. So it goes, it grows and descends in the bone, forming larger cells here, which becomes hypertrophied and enlarged. And when they reach their maximum growth here, actually the other osteoblasts surrounding or nearby here will start secrete the osteoid matrix, which gradually surround these cartilage cells. The chondrocytes will die, actually. They will die by apoptosis or programmed cell death. At this point, the osteoblast will take their place, they will replace these dead cells and live here and start forming the bone tissue. The bone tissue finally will be formed here at this area in the form of a regular bone or spongy bone, which later on will be remodeled and will be transformed actually into compact bones. Let's talk about the mechanism of calcification. What about how this bone, the mechanism of calcification occur? Exactly. The process is not completely understood. But the osteoblasts secrete a vitamin K dependent polypeptide, which is called the osteocalcin. Osteocalcin, together with glycoprotein, will bind the calcium ions and raise its local concentration. Osteoblasts also release alkaline phosphatase, which raise the local concentration of phosphate ions. The formation for this will be followed by the formation of hydroxyapatite crystals. These crystals will grow rapidly, forming a mass of calcified material with collagen and proteoglycans, giving the hardness of bones. Okay? So this is generally the process of formation. What about the process of remodeling? Remodeling, reshaping the bone. Okay, and this process is greatly facilitated by the osteoclasts. The osteoclast will absorb the matrix of bones, so eroding sides and edges and surfaces of the bone, giving its final shape. And this is what is meant by bone remodeling. And actually, remodeling is done by two sides: a side of destruction or absorption, and the other side of bone building. The bone remodeling is a combined process of bone synthesis and removal. And normally, about 5 to 10% of our bones, of adult bones, turns over every year. So resorption occurs by osteoclasts, and the reabsorbed areas becomes invaded with osteoprogenitor cells and blood capillaries derived from the industrium or periosteum. The osteoblasts develop and form the concentric lamellae of bone and becomes trapped and transformed into osteocytes. See, this diagram will show you the bone processes. A process of bone growth on the left side and the process of remodeling on the right side. See this dashed line here, the red. So this is the bone will be formed by remodeling and removing this surface either by reshaping the bone. The remodeling means reshaping the bone, okay, by two processes working together. So what about the process of bone growth? The bone growth is different from, is a different expression from bone ossification. This is how the bone becomes bigger, how the process of growth. The growth here, there is two types of growth, like cartilage, a positional growth. Here, the positional growth. How does it occur? It occurs by two processes. The process of osteoclast remodeling. The osteoclast here will absorb the industrium from the inside. It will eat it. And the osteoblast from the outside in the periosteum will lay down more layers here on the outside. So the bone diameter will be enlarged gradually from the infant to a child to the adult. And this is what is called a positional growth. Type. What about the factors influencing or affecting the bones? Of course, bones, as we said, is affected or influenced by parathyroid hormone, which transform the bone calcium into a blood calcium. On while the calcitonin, the thyroid hormone, calcitonin, do the opposite, which means that it takes the calcium from the blood and put it in the matrix of the bone. So it resists rarefaction or lowering the bone calcium. The calcitonin, that's why calcitonin is used for treatment of osteoporosis. Deficiencies in blood calcium or phosphorus leads to bone rarefaction and osteoporosis and more liability to fractures, which are called pathological fractures, in fact. Deficiency in vitamin D will lead to disease, a famous disease known as rickets, while deficiency in vitamin C will lead to scurvy, and radiation may destroy bone cells and other factors. Drugs may do also a similar role or similar effect. Calcitriol, in fact, is the physiological active form of vitamin D that mediates its biological effects by binding to vitamin D receptor, which is abbreviated by VDR. The VDR is the vitamin D receptor, which is principally located in the nuclei and the DNA of the target cells. Vitamin D, the binding of calcitriol to the VDR, allows the VDR to act as a transcription factor that modulate the gene expression of transport proteins, which is responsible for the first step in calcium absorption in the intestine. That's why vitamin D is very important for bone growth and ossification. It enhances the absorption of calcium in the intestine. Let's see the hormonal mechanism and the hormonal factor that act on the deposition of calcium in the bone and the resorption of calcium from the bone and return back to the blood. The circulation of calcium from bone to the blood and from blood to the bone. Here, the two antagonistic effects of the calcitonin and the parathyroid hormone. Let's see if here if there is a decline in the blood calcium here will stimulate the thyroid gland to what? Blood calcium is lowered. So we need more calcium. How do we get the calcium? By releasing parathyroid hormone, which stimulate the osteoclasts of the bone to dissolve, to release the calcium from the matrix and then raise it back to the blood, which will stop the parathyroid hormone from working and back stimulate the thyroid gland to release the calcitonin, which will stimulate the calcium salt deposition in the bone. So both hormones are working with against each other and in fact are complementary to each other to make or to do the necessary balance of calcium and the hardness of bones. Here, the falling of blood calcium levels signals will stimulate the parathyroid gland to the release the parathyroid hormones. The parathyroid signals the osteoclasts to degrade bone matrix to release the blood calcium. The two hormones are actually working against each other, but doing the necessary balance for keeping the bone structure and function. By this slide, I think I have finished today's lecture about bone and ossification. I thank you very much for your listening, and I hope you enjoyed tonight. And thank you for listening, and goodbye.